Well, hello, good evening, everyone. So let's not waste more time by waiting for the participants to join. We'll continue while the others keep joining. So before I start with the introduction of our scientist here, let me just introduce you to all Nakshatra. So yes, Nakshatra Sciences and Collaborative Research is a community for researchers and by researchers. So Nakshatra Sciences was established around a couple of years ago, and today we are a people of almost 25,000, both offline as well as online. And speaking more about it, we are a project-based community learning, and we have our physics wing well established at Nakshatra already. So life sciences, we are just keeping our baby steps now. So hope to see in building a community of biologists here. So that's the main purpose of having been hosted about Nakshatra and telling about it. So also it's a nationwide community, and we have provided programs for UG students, working professionals, as well as teachers. And we enable undergraduate students and above to solve like real world problems by connecting them directly with industry experts and the research community. So why we do this? This is because there is an students, intermediate need for skill training programs for research and industry. So there's also, if we could see that there is an inaccessibility to good opportunities in almost many domains of science, especially in STEM. So, well, that's a bit about Nakshatra. We have always had a few young scientist programs for kids, and then we have programs for undergraduation and above. And yes, we have made offline programs as well as online programs. And we have a panel of over 100 mentors, scientists, and researchers from around the world. And also, we have conducted over 100 events in the span of 2 years now. So presently, we are a part of 20 colleges and almost 5 schools. We, as Nakshatra Sciences, we focus mostly on creating learning ecosystems and focus on experience building, as well as hands-on programs and projects. We encourage scientific thinking. So that's about it. So this was a brief about Nakshatra Sciences, and talking more about our life sciences wing. So as I said, that physics wing is well established. It's already going with 3 projects currently, and life sciences wing is new. We have successfully completed our workshop earlier named Xylem Workshop. It was a certification program wherein we invited scientists from all over the globe to give us lectures and guest talks, thereby introducing people to biology and its world. So Xylem workshop was a successful one, and now we have taken a little bigger step in getting up our new project named Molecular Dynamic Simulations, Visualization and Analysis of Biomolecules. So in support of the same, today we have our scientist Dr. Shashanka K. Prasad to actually go through our next upcoming project in supportance to computational biology. So yes, that was about our brief introduction for Nakshatra and what it does. So furthermore, I would now take up this opportunity to introduce our scientist and the guest lecturer for today, Dr. Shashanka K. Prasad. So well, hello everyone and hello sir, officially. So let me just introduce him. Dr. Shashanka K. Prasad has completed his integrated course of 5 years, MSc in Molecular Biology in Yuvaraja's College, University of Mysore. He has completed his PhD in Biomedical Sciences from JSS Medical College, JSS Academy of Higher Education and Research, Mysore. He has been elected as a Research Fellow by the Linnean Society of London, United Kingdom. He is also a MRSB membership from Royal Society of Biology, UK. He is also an Associate AMRSC from Royal Society of Chemistry, UK in the year 2022. Dr. Shashanka is a postdoctoral research fellow from the Faculty of Agriculture, Chiang Mai University, Thailand. He is currently pursuing his postgraduate diploma in intellectual property laws from National Law School of India University, NLSIU, Bangalore. He is also an ambassador and early career researcher at European Association of Cancer Research. He also extends his service at Society for Medicinal Plants and Natural Product Research. He is also a professional member from International Association of Oncology. Dr. Shashanka holds 2 academic board membership, member of Board of Studies and member of Board of Examinations, Department of Biotechnology, Bharatiya College, Mandya, affiliated to University of Mysore. Dr. Shashanka holds his further achievements in the editorial and research guidance section. To name a few, he has served as an assistant editor in the International Journal of Health and Allied Sciences. He has also served as a review editor in journals such as Frontiers in Microbiology, Frontiers in Bioengineering and Biotechnology, Frontiers in Environmental Sciences, etc. As a part of the Young Science Leader program, Dr. Shashanka has mentored over 26 students across India in various disciplines of life sciences. Furthermore, some of his collaborations involve Wasaf University of Life Sciences, Poland, Chiang Mai University, Thailand, University of Nottingham, Malaysia campus, University of Padjadjaran, Indonesia, Kansas State University, USA, King Saud University, Saudi Arabia. He has presented a number of posters and presentations in India and abroad. He has been a part of 11 FDP programs in biotechnology and bioinformatics. He has a continuing education program on data analytics and predictive technologies from IIT BHU, India. He extends his versatility in 9 conferences and workshops in various domains of life sciences. Dr. Shashanka has 2 patents filed in his name, more than 40 research papers and articles in well-known journals. He has 3 book chapters under his name. Currently, Sir has been working as a researcher in Chiang Mai University, Thailand. And this evening, we have him on board with Nakshatra Sciences and Collaborative Research to deliver a guest lecture in describing the beautiful confluence of biology and computers in support of our upcoming project titled Molecular Dynamic Simulations, Visualization and Analysis of Biomolecules. Over to you, sir.
Thank you, Dhanushri. And I take this moment to thank Nakshatra Sciences for this opportunity. And I wish them all the best for their future endeavors. And now, let me share my screen. Please let me know if the screen is visible to all of you.
Yes, sir. The screen is visible.
Yeah. Okay. So I think we should start with today's lecture. And I am here to talk about the confluence of biology and computers. This topic, where you have the confluence of biology and computers, it is something very vast, and there is a lot of growing research and growing advances in this particular field. So let us try to understand what is the confluence of biology and computers like, and what exactly is the outcome of such confluence. And in addition, I'll also possibly let you know about the scope for such a branch of science. So to begin with the introduction of this particular topic, why do we need computers in biology? Or what is the purpose of having computers in biology is the first thing that comes in our mind. And when you say computers in biology, it is not limited to the usage of the Microsoft Office or any other tools, but computers can also be used in biology for the data analysis, mathematical modeling, and computational simulations to understand the biological systems and relationships. So the moment we say simulation, this is a particular term that keeps coming across this particular talk. And in order to not confuse you people, let me tell you the typical meaning of a simulation. Simulation, according to a dictionary, means that it is just an imitation of an already existing circumstance. So the moment you say computational simulation, it is the imitation of something biological that is being imitated or that is being put up on a computer. So getting more into the details, like this particular concept or this particular point where computer science and biology meets is what we call in the layman terms as bioinformatics. But it is not just a combination of computer and biology, but there is also mathematics and statistics involved. And all of this together with the use of the big data generated out of the information engineering will constitute the entire topic or entire syllabus of a bioinformatics topic. So, moving on further, what are the applications of such a Confluence? See, in biology, there are many things that you can do with regard to bioinformatics. And first thing is bioinformatics itself, and where the bioinformatics as a application will involve the informations of biological components or biological data. Okay, so this can be interchangeably used when you're talking about bioinformatics as a particular subject. It comprises of all these applications, but when you're talking about bioinformatics only as an application, it is the information systems of biology. So apart from this, what can you do? There is, you can understand the biomolecular interactions. You can understand how a biological network works. You can analyze DNA. You can carry out comparative genomics, where you can compare the genes or the genomes from two entirely different species or organisms and find out the similarities between them. You can also do what is known as the drug development, and which will involve both the drug designing and also drug discovery. Then there is health informatics, where you can store the health related data of people. Nextly, there is sequence alignment, which is a type of comparative genomics or DNA analysis, where you compare the DNA sequences side by side and check for their alignment with each other. Furthermore, you can simulate the cell division process. You can carry out pattern recognition, which is an essentially a machine learning concept. Then you can do gene finding, where if you have a very long sequence of DNA, you can use the computer to find genes, which can eventually get translated to proteins. Then nextly, we have the phylogenetics, which is the evolutionary understanding of how these macromolecules, be it the DNA or the proteins, and eventually the organism itself, how they evolved over the period of time. And then you have the functional genomics, which gives you an understanding of how the gene expresses itself and how you can possibly knock it out, or how you can reduce its expression, and how this kind of an expression will have an impact on the health system. And then we have the computational biology, which is a way of how you understand the biological systems using the computer. And finally, you have the metabolism, where you check for the different metabolites that are produced in the body and how they react. So all of this can actually be done using a computer. So getting into the details of the things I just told you, with the specific reference to my expertise, I would like to tell you that you can do the DNA sequence analysis in the form of BLAST, or you compare two different sequences next to each other, and that is a local alignment. And then you have the clustol, where you do multiple sequence alignment. So there are multiple sequences that you take into consideration, and you check for the alignment. You can also do promoter analysis, gene prediction, analysis of regulatory elements, the identification of the introns and exons in the gene or in the DNA sequence, and the primer designing, which can help you in your PCR experiments. Then you can also do the codon usage optimization and virtual translation, where here in this case, you can literally obtain the protein sequence from a gene sequence. So you know that any gene sequence will have a lot of other information apart from what can go into a protein. So with the help of intron and exon finding, you can identify the exons, and then the next step will be the virtual translation, where you can get the accurate protein sequence that can be obtained. So moving on to the next or other kind of application, there is the molecular dynamics simulations, where you can visualize computationally the protein DNA interaction or the protein ligand interaction, and also the drug DNA interactions. You can carry out drug designing or drug development, where you can do a plethora of things like the identification of protein targets and validation of the targets. Then you have the lead identification, where the lead will be that molecule which is a possible drug that you are evaluating, and you optimize this particular drug for to improve, possibly to improve its activity or to improve its efficacy. Then we also have the ADMET prediction, that is absorption, distribution, metabolism, excretion, and toxicology prediction, where this drug molecule, the lead molecule that you have here, you check for these properties of this particular lead molecule to make sure that this is a potential drug. Now, moving on to the proteomics part of it, you have the protein sequence analysis that you can do. You can check for the molecular mass of the protein sequence. You can check for its isoelectric point. You can check for the instability index. You can identify the amino acid composition. Apart from this, you can look out for the domains and carry out motif search. And you can do the signal peptide identification, the secondary structure analysis, etc. Moving on to the evolutionary analysis or the phylogenetic analysis, you can reconstruct a complete evolutionary history of even if it is a small sequence of DNA that you have, or even if it is an organism, you can completely reconstruct, and you can find out the ancestors of that particular organism, and also the ancestral sequences of that particular DNA or the protein. Then you can check the gene flow. You can analyze how or at what point of evolution a particular gene came into the system and how it got modified to meet the required conditions, etc. And you can also identify the conserved regions of the DNA which have remained as a part of that particular sequence across the ancestors. Then we have the molecular interactions, where you can check for protein-protein docking. You can identify inhibitors and activators of proteins. You can look into the protein DNA interaction. You can also check for the transcriptional factors present in the system, and you can also look into the protein ligand interaction. And lastly, you have the molecular modeling, where you carry out the 3D structure prediction, wherein if you have a protein sequence or a DNA sequence, you can literally predict the structure of the protein. And also, by the virtue of this structure prediction, you can also predict the function of the protein. So this is one example of where what are the different ways in which you can identify a mitochondrial DNA related disease, or how to address it with the help of computational biology. There are different aspects which we call as the omics with regard to bioinformatics. Omics means the study, and omics of the genes will be the genomes, or omics of the genome will be the genomics. And omics of the transcriptomes, or the transcripts, or the mRNA, will be the transcriptomics. Then we have the epigenomics, which deals with the epigenetic interactions. Then we have the metabolomic of the study of the entire metabolites which are produced within a cell. Then the proteomics for the entire set of proteins produced within a cell, and the microbiomics, where you look into the microbiomes of an individual. So you take all of them into consideration, or any single one of them into consideration from based on the biological sample. Then you do some analysis using a computer, and then using this multi-omics approach, you can improve the patient health. You can get an insight on the mitochondria, and you can also improve the diagnostic and therapeutic aspects to create what is known as the personalized medicine, which is best suited for that particular individual. So this personalized medicine is what we know today as the pharmacogenomics. So now moving into the most important aspect of bioinformatics, they are the databases. Databases are a collection of libraries. I'm sure all of you have visited a library at some point of your life. And in a library, you can see that the books are systematically organized depending on the subject, depending on the language, or depending on their level of advancement, etc. Similarly, database is a systematic arrangement of such libraries. So when you say a database, it is not something small, but it is something associated with the big data. And with regard to bioinformatics, databases, according to the Wren and Bateman, are the stores of biological information. And there are a wide range of databases that are available today. These are a few important databases that I have just informed here. We have the NCBI, which is the most famous database, which I'm quite sure all of you would have come across by now. Then we have the EMBL, the DDBJ, or the DNA Data Bank of Japan. And all of them in collaboration, they have the nucleotide sequence database. And then we have the PubChem, which completely deals with the chemical components or the molecules that you can find in chemistry. You get the structural details, and you get the details of the physical and the chemical details of any kind of a drug that or a chemical molecule or a metabolite that has been reported already. Then we have the PubMed, which is a database of articles. We have the KEG, that is the Kyoto Encyclopedia of Genes and Genomes. You have the Human Metabolome Database. We have the ProCite. ProCite is by the SIB, that is the Swiss Institute of Bioinformatics, which also runs the SwissProt. Then we have the Protein Data Bank, which is essentially the go-to place to get the protein sequences and protein structures. And we also have a lot of databases that are specific for model organisms. And one such example is that of the fly base, which is a database for the Drosophila melanogaster fly. And these are the most important databases that you come across on a regular basis. We have the GenBank by the NCBI, which has the nucleotide sequences. You have the Ensemble, which has got the human or mouse genome, and also the other model organism genomes that have been reported so far. You have the PubMed for literature references. PubMed is by the NCBI. And then you have the non-redundant database, which is by the NCBI, and which has got the entire information of the non-redundant protein sequences, meaning the proteins which are essential for any organism. You will possibly find it in the non-redundant database. Then we have the Swiss Prot by the Swiss Institute of Bioinformatics for protein sequences. Then there is Interpro by the European Bioinformatics Institute, which is a database for protein domains. Then there is the OMIM, that is the Online Mendelian Inheritance in Man, in the NCBI for the genetic diseases. You have an enzymes database for the enzymes. And there is the Protein Data Bank for protein structures, and KEGG for metabolic pathways. So giving you an input about a particular database on how exactly a database works and what can be found in a database, I would like to take you along this path with an example of NCBI. Personally, if you ask me, I would say NCBI is like a database of the databases. It is like very vast. And it has got multiple databases that you can access and you can utilize throughout your academic or research career. So the NCBI databases are broadly classified into 6 groups. You have the literature, you have the genes, you have the proteins, you have the genomes, you have the genetics, and you have the chemicals. So under literature, you have the first thing that you can see here is the bookshelf. Bookshelf is a database of books and reports. So if you, as a student or as a researcher, if you're looking for some kind of a book that you want to refer to, I think you should try and visit the NCBI bookshelf, and you can literally browse the titles, and you can search the books with the help of the new releases. And also, if you're an author or a publisher, you can participate by contributing your information to this particular database. Next, there is the PubMed and PubMed Central. PubMed is for the scientific and medical abstracts or citations, whereas PubMed Central, which is a subsidiary of the PubMed, can give you access to the full text journal articles. So this is what the homepage of PubMed looks like, and this is maintained by the National Library of Medicine. And the National Library of Medicine, in fact, maintains the National Center for Biotechnology Information, which is nothing but the NCBI. And here, if you just look for any particular topic, like here in this case, I have searched, I had just given one search command of bioinformatics, and I got a lot of results, as many as 69,074 results only for bioinformatics. And that indicates that they have as many as 69,074 articles for bioinformatics in the PubMed. And you can also individually filter based on your requirement. If you are looking for a free full text article, you can select this. If you're looking for something latest, something that happened in the year 2020, you can click here. You can narrow down your search. And then if you have anything particular in mind, like if you're looking for a clinical trial, you can do that. If you're looking for any kind of a review, you can select here. And depending on the publication date, if you're like usually what happens is whenever you're writing an article or a thesis, it is generally advised that you stick on to the most recent publications within the last 10 years, unless the publication that you want to cite is of utmost importance. So you have a filter for the publication date as well. Next, we have the database for genes, where you have the gene sequences and annotations, which are used as references for the study of orthologs, structures, expression, and evolution. So when you look into a particular gene, the gene I have chosen here for this example is the Homo sapiens SRY gene. I hope you all know what is the SRY gene, or just in case you don't know, this is the gene that is the most crucial point which determines if an individual or a human being is going to be a male or a female. We'll talk more in detail about this gene in the coming slides. So here, when you do any search for any gene in the GenBank database, remember there is always an accession number that is there, and always make note of this accession number. Wherever you go, the accession number is the most important information, because you have a very large or very wide range of results that you get for a single gene. So in order to avoid confusion for you, always make a note of the accession number. And here, this will be the first page when you open the particular gene. And there are different ways in which you can view the information. The genetic information is here in this particular page, where you get the locus of the gene, where exactly it is present, how big it is, and who are the authors who published this particular information, and everything about the particular gene is available here. But as a genomic individual or a person who is doing genomic research, what is the most important is this particular format, which is known as the FASTA format, where you get nothing except the gene sequence. And in most of the cases, the FASTA format is what you need for further analysis. Or if you're doing a normal genetic study, you can also use the graphics to get the graphic output of the sequence that is available here. So now there is OMIM, that is the Online Mendelian Inheritance in Man, which is basically a database of diseases, particularly the genetic diseases. And it is maintained by the National Institute of Genetic Medicine, the Johns Hopkins University School of Medicine. And it gives you the complete information on human genes and genetic phenotypes that are updated on a regular basis. So for the same SRY gene, if you look into the OMIM itself, you get a wide set of results. Like you have as many as 101 results here. And the SRY is the short form for sex determining region Y, because it is found in the Y chromosome. Alternatively, it is also called the testis determining factor, or the TDF, or the testis determining factor on Y, the TDY. So here you get a different output or a different way in which you can look into the gene and in the OMIM. So you have the phen gene graphics, which help you visualize either linearly or radially the relationships between the phenotypes, the groups of related phenotypes and also the genes that are associated with the particular gene of interest that you're looking into. So this, when you choose a linear representation of the gene that you're studying, you can get a lot of information on how exactly the gene flow happened and who were the exact ancestors and what are the exact genes that have been reported so far, and what was the outcome in the form of the SRY that you have here and that you're studying. Additionally, this can also give you a link to numerous external databases. When you say database, it is not a small thing, it is a very big thing, and it can give you link to numerous databases. Like, if you want to study the genome, then there is the NSimple database, the NCBI genome database, the viewer, the University of California SE genome or database. Then you have the genome browser. Similarly, you have the DNA database, you have the protein databases that you can be linked to. You have for the gene information, you have a variety of databases. And if you want to look into the clinical resources associated with the SRY gene, you have linked to that. Then if you want to check out the variations of the SRY gene, you can study them here. Then if you want to understand on what kind of a research has happened on the animal models with regard to the SRY, you have linked to that. And additionally, you have linked to what is known as the Reactome database to understand the cellular pathways which involve the SRY gene. So now, moving on to what you can do with such databases, with such large amount of information, how you can implement them, or how exactly this information can help you with your research. So the first thing that you can do is an alignment. Alignment is where you can make use of the BLAST. That is the basic local alignment search tool, to find the regions of similarity between biological sequences. And this, understanding of regions of similarity will help you understand on how this protein or how this gene behaves across the organisms. Like if you have a particular gene that is present exactly the similar way or the same way in a mouse, then it is understood that this gene is possibly behaving the same way in you as it behaves in a mouse. And in order to understand such things, you can do a basic local alignment search tool. There are 4 different types of BLAST. First is known as the N-BLAST or the nucleotide BLAST, where you compare a nucleotide with a nucleotide. The next type is the P-BLAST, where you compare a protein with a protein sequence. Then there is something called a BLAST X, where you have a DNA sequence, but you want to compare it against a protein. So you can use BLAST X to translate this particular nucleotide sequence into the protein and analyze the alignment. Or you also have the T-BLAST N, which is the reverse of a protein to nucleotide alignment analysis. So here, just to give you an input on how exactly a sequence alignment looks, this is a multiple sequence alignment. You have same DNA sequence from different organisms. These are all plants. And as you can see, you have 13 different plants. And whenever there is an alignment that is appropriate, you have a particular color code. And if there is a change in the alignment, as you can see here in the case of C. sativus, that is highlighted, or if there are no nucleotides of this particular code available, then you have the blanks that are left out across the study. And this gives you an idea of how a particular gene would look like in a particular organism. But this is the multiple sequence alignment, whereas this you see here is a, local alignment or a pairwise alignment. So now let us try to understand with an example of cancer how you can use this information. There are a variety of things you can do with this particular bioinformatics database that you have. And I've chosen cancer because that is in line with my area of expertise. Since I am a cancer researcher, I can tell you more about what I can do with regard to cancer using bioinformatics. So firstly, there is a particular database for cancer, and that is known as the Cancer Genome Atlas. That is the TCGA. This is a component of what we call as cancer genomics, where genomics, as you know, is the study of the sequence of those nucleotides in the form of letters that are present in the DNA, and how each string of these nucleotides pass on the information to help the bodily functions. So if there is any single mutation happening in any of these letters or nucleotides, then they will lead to the formation of what are known as the mutated proteins, which can make the cells grow quickly and cause a damage to the adjacent cells. And this is essentially what we call as cancer. So the moment you start understanding the cancer genome, it will be very helpful for you to personalize medicines with the help of what we call as the pharmacogenomics. And this will help in the selection of the best treatment for each patient, because every cancer is different from the other one. You have trillions of nucleotides in the body. And out of these trillions, at least a billion of them, or maybe a million of them, translate into different types of proteins, like tens of thousands of proteins. So the mutation is not necessarily uniform in every type of cancer. And for you to treat cancer, you should understand the kind of mutation that that particular patient or individual has gone through. And that is where the cancer genomics or the cancer genome atlas comes to help. So this is a database maintained by the National Cancer Institute, or the NCI USA. And it has got information of over 20,000 primary cancers, which is spanning 33 different types of cancers. And also, this is a publicly available resource, and anybody can use it in order to, understand on what is cancer, what are the cancer statistics, what are the disparities with regard to cancer, how to treat cancer, what are the side effects, what are the clinical trials that are being carried out currently. You get up-to-date information about the different types of cancer drugs, how they behave, what are their interactions like with the normal systems. And also, they have a particular or a dedicated database to tell you about the complementary and alternative medicine. With the advancement in science, there is also a growing prominence for the traditional knowledge in the form of Ayurveda, Unani, Chinese medicine, etc. All of them are regarded as alternative medicine. And you have information on alternative medicine as well, and not just the allopathy or the English medicine, as we can call it. It can help you understand on how to cope with cancer, like just in case you are, taking care of somebody who is suffering from cancer, then you can also be regarded as a caregiver, and you get complete information about how to deal with cancer as a researcher, as a caregiver, and also as a common person. And furthermore, you have cancer specific details where you can get every up to date information about all the types of cancers that are there and that haunt the humankind. So this is like, so far over like 11,000 tumors have been analyzed, and this visualization that you see below, it gives you a glimpse of what is known as the pan-cancer atlas. That is a series of shaded rings that are joined together to create a beautiful singular spectrum. Okay, so this is where one type of cancer research or is merging with the other type, and with the different types of cancers you have, this is the kind of output that we have got today. And we have possibly come to this extent and understood this in the form of this pan-cancer atlas only with the help of computational biology, where you can literally reach out to anybody in the world and understand on what a researcher in America is doing by sitting just in India or Thailand or wherever you are. So there are different types of computational tools that are provided by the TCGA. You have the Cancer Imaging Archive that provides you access to the radiological imaging data. You have the Cancer Proteome Atlas, which gives you access to the proteomic data associated with cancer. We have the CBio portal. Then there is the copy number portal. And as you see here, this is the network visualization of the cancer genomics data in total. So these are the different types of tools that are available. You have the TCIA, that is the Cancer Imaging Archive, maintained by the NCI. You have the TCP, or the Cancer Proteome Atlas portal, maintained by the MD Anderson Cancer Center. The CBio portal for cancer genomics by the Memorial Sloan Kettering Cancer Center. The copy number portal by the Broad Institute. The DMIXT by the MD Anderson Cancer Center. Then we have the FASMIC by the MD Anderson Cancer Center. Then there is Firehose and Firebrowse by the Broad Institute. We have the FunSeq2 by the Weill Cornell Medicine. The IGB, or the Integrative Genomics Viewer, from Broad Institute. There is MBatch by the MD Anderson Cancer Center. Muse, or the mutation calling using a Markov substitution model for evolution, by the MD Anderson Cancer Center and the Baylor College of Medicine. Then you have the Regulome Explorer by the Center for Systems Analysis of Cancer Regulome, and the ServNet from the MD Anderson Cancer Center. Then there is Standric by the MD Anderson Cancer Center. The Tumor Map, which provides a high-dimensional omics data in a two-dimensional projection, by the University of California, Santa Cruz campus. And the Xena by the UCSC. Next, we have this other beautiful database called the String. String here stands for the search tool for retrieval of interacting genes or proteins. So this is a biological database which will help you understand the protein-protein interactions. So I would like to explain this with an example that I have chosen. This is the String homepage, and here you can search for any particular protein, and I've chosen the BRCA1 protein, which is a breast cancer protein found in Homo sapiens. You will have different types of organisms that will have different types of genes, and you can literally search for them, for any of them. So the moment I gave search, there were several matches that came up, and my gene of interest was the BRCA1 gene, that is the breast cancer type 1 susceptibility protein producing gene. Uh, and that is the protein that we have here. So I select that. And now when you look into the output, you have the BRCA1 protein here, and this is what the protein looks like. And these are the different other proteins that are interacting with the BRCA1. So when you are into cancer research, BRCA1, as I told you, is specific to breast cancer. And when you are into cancer research, you may want to understand on how different proteins are impacting that particular cancer. So for that, you should know the proteins involved. And the moment you run a string search, you get the result like in this form. And if you want to know what exactly each protein is, I have chosen the barred one here. And when you click on that, you get the complete information about the protein. So this barred one is the BRCA1 associated ring domain protein. And you get all the details about the protein, and also you can take it forward for further analysis. So talking about taking a protein or a DNA sequence forward for further analysis, we have what is known as visualization. Visualization of macromolecules is a crucial point or a crucial part of bioinformatics. And as you can see here, you can literally, if you just have a particular sequence, you can get how this particular protein would look like, or a DNA would look like. And also, as a part of visualization, you can get into the details of how these proteins behave in a physiological system. So here, as you can see, for an example, I have given you a protein sequence, and this is a particular a drug molecule. So with just these two information, with the help of computational biology, you can predict the protein structure. And also you can check how this particular drug molecule is interacting with the protein, and what are the different amino acids present in the protein that are interacting with the drug molecule. So as a part of visualization, you also have a variety of tools. You have the UCSF Chimera, which is a very important tool where you can also carry out the protein standardization. And then there is also UGene, which can be used for both the DNA sequences and also the protein sequences. Nextly, we have the PhyLab for the visualization of phylogenetic trees, and to understand how exactly the evolution could have occurred, and the possible common ancestor between different organisms or genes or proteins. Next, we come to another very important application of bioinformatics. Bioinformatics today is essential for computer-aided drug development. So you can literally develop drugs in computers, check out how they work, screen them, and then take them for further analysis. And this actually is a very fast growing technology. And today, during COVID, in the last 2 years, there have been numerous research that have been carried out with the help of CAD. And CAD is an essential tool with regard to bioinformatics. So what happens in CAD is, like, as I told you earlier, you have the target selection and target validation that you carry out, where you do identification of the proteins. Then you check on how this particular target interacts with different types of molecules to identify that one or a few molecules which are possibly the candidates that can interact with the protein and stop or help in stopping the disease. So these molecules will be called the lead molecules. So you can carry out the lead discovery, and once you discover the lead, you can carry out the lead optimization to make this molecule work better. Or if this molecule is a toxic molecule, you can do lead optimization to reduce the toxicity of the molecule. And then there is a preclinical test that is done that can be done in silico with the help of the ADMET prediction, that is the absorption, distribution, metabolism, excretion, and toxicology prediction. So this essentially will help you know what kind of a modification this particular molecule can undergo once it gets into the biological system, once it gets into the human body via the oral route. And then you also have the pharmacokinetic simulations and computational system biology analysis that you carry out before the clinical trials. So just to give you an idea of how demanding the drug discovery process is and where exactly bioinformatics fits in. Bioinformatics plays a very important role in the first 3 years of drug discovery, or the first 3 stages of drug discovery, where you use bioinformatics to gain the knowledge about the disease, to gain knowledge about the different proteins that are involved in the disease, and to gain knowledge about different molecules which can be potent druggable compounds against that particular disease. Then you do target validation to identify the lead, and then this lead molecule is validated further to check for its efficacy. So until here, bioinformatics is very essential. And in fact, it is a growing area of science which is taking over the conventional methods. And once you have identified a lead molecule, there is a preclinical development that is carried out, preclinical development analysis. And then you move on to the clinical analysis, where you check if the drugs are safe in the humans. And this happens multiple times, and then the molecule is out in the market as a drug. And despite the sale of the molecule, there will be a phase 4, which is known as the post-marketing surveillance, where the molecule is constantly monitored for its efficacy and also for its impact on the human life. And you read regularly in the newspapers saying, like, the World Health Organization banned so and so drug from usage and things like that. And that is mainly because as an outcome of the post-marketing surveillance. So this is the entire drug discovery process. And when you do a computer-aided drug development, there are two ways in which you do it. First is the ligand-based drug design, and second is the structure-based drug design. In the ligand-based drug design, you already have a ligand. Ligand is the drug molecule. And ligand is something that interacts with a protein. So you already have a molecule, which you call the ligand. You carry out some quantitative structure activity relationship studies. You understand the pharmacophore of the ligand. You do similarity searching to identify similar molecules. Then there is scaffold hopping, where you screen all these molecules with similar structures. And then you make use of machine learning to identify the best ligand that is available for interaction with the protein target. And then you can also do a ligand-based virtual screening to identify the best ligand interacting with the protein. So the advantages of the LBDD, as we can call it, is like, it can proceed without a protein structure because you already know that there is a ligand and this ligand interacts with the protein. So you can develop a lot of predictive models, and this technique is less complex compared to the SPDD, or the structure-based drug designing method, and it also requires a very low computational requirement as compared to SPDD. Moving on to the limitations of LBDD, it will be difficult to determine the bioactive conformation of the structure of the ligand, because none of them are static. The protein is not static. You will understand that in the slides to come. And the ligand is also not static. So it will be difficult to determine the bioactive conformation due to this dynamic nature of the ligand and the protein. And there are also pharmacophore constraints that you have to take into consideration. And also, the methods are generally ligand biased. So moving on to the structure-based drug designing, here you develop a structure, you develop a new molecule, and you carry out molecular docking. Then you carry out the molecular dynamics simulation, and then that will be known as the structure-based virtual screening. The advantage here will be that the protein structure will also be used. As you can see here in this image, you only see the ligand, and the ligand is taken up for analysis. Whereas here you see a particular structure which is chosen as the ligand and also the protein. So you understand the protein ligand interaction, and this is a ligand unbiased method. So you can determine the bioactive conformation, and you can optimize the ligand depending on the requirement of the protein. But there are certain limitations where you might not really get a proper pose versus score correlation. It might be difficult to predict the correct pose, and also you should acknowledge the protein flexibility, and you won't really know, since it's a computational simulation, you won't really know the exact flexibility of the protein and how it behaves within the biological system. So there are different tools that can be used for the CAD. We have the visual molecular dynamics software, that is the VMD, which I was told by the Nakshatra group that they use the VMD here. And also there is a very, very expensive software called the Schrodinger. I personally work on Schrodinger. Like we have purchased from the JSS Academy of Higher Education and Research, where I work. We have purchased Schrodinger, and this particular software costed us as much as 3.5 crores. But Schrodinger is like, the applications of Schrodinger is like very extensive. Yeah, it is not limited to drug discovery, but I'm sure like you can apply the same principles in all the other softwares as well, depending on your requirement. Then there is Autodoc. Autodoc is a free tool, and it helps you in docking. And there is the Discovery Studio by the BioVidia. We have the Molecular Operating Environment, or the MOE. Then there is the SEED, which is the short form for Solvation Energy for Exhaustive Docking by the University of Zurich. And then there is GROMACS. In my knowledge, the molecular docking is carried out by all of them, but a few of them, very few of them, like the GROMACS, the Schrodinger, and the VMD, and also the Discovery Studio, can help you with the MD simulations. So what are MD simulations? I'll tell you now. So whenever you are doing a drug discovery process, you have a protein and the molecule of interest that you're analyzing, and if this molecule has very good conformation with the protein, then it has to go through certain analysis for the drug-like properties. So here in the drug-like property analysis, you have three different analysis that you do. Firstly, you check for the Lipinski rule alignment. This rule is also known as the rule of five, or the drug likeness analysis. And then you check for the ADME properties, and finally the toxicity assessment. So both these articles are for COVID, or they were trying to identify drugs for COVID using bioinformatics. Here, I'll give you an example here, and you can correlate with this particular flowchart as well. So what can be done is, you prepare a protein and you create a ligand library containing different ligands. And then you carry out docking study to this complete ligand library against the protein target. And then you identify the lead molecules, which we call as the hits, by carrying out an interaction analysis between the protein and the ligand. And then you estimate the binding strength, which will tell you on how good this particular lead ligand that you are choosing is interacting with the protein. And then you move on to the drug likeness analysis and also the MD simulation, or the molecular dynamic simulation analysis, before reporting a particular lead compound as possible druggable or drug molecules. So this is what a Schrodinger output would look like. This is a three-dimensional interaction. You can see the bond between the protein and the molecule here, and this is a two-dimensional interaction which shows the ligand structure and also the different amino acids that are interacting with the molecule. And also here you have the docking score, which is nothing but the binding affinity binding affinity of the this particular protein ligand interaction, etc. So in this animation, as you can see, you have a single protein, which is a static protein, not dynamic, but you have different molecules that are being screened to identify the best fitting molecule that can fit into the binding site of that particular protein. So this is how this is an animation of how the docking works. Next, once this docking is done, remember the protein is not flexible here, but in a biological system, the protein is a flexible molecule. So your docking result might not be accurate because the protein flexibility is not included. So in order to confirm this docking result, you have to carry out what is known as a molecular dynamics simulation, where you simulate the physiological system and and then you check how the protein also changes its conformation, and also how the ligand molecule also will change its conformation to stick onto the protein. So the MD simulation is a very important output that you need to get before you say a particular molecule is a possible drug candidate. This was one of our results that we published in Cancers recently. So we did a virtual screening of Moringa oleifera phytochemicals against the MAGA genes for the non-small cell lung cancers. So there were two aspects here. Firstly, we did docking, and also we got a two-dimensional output where we saw that one particular phytochemical was interacting really well with the protein, and it was attaching to different types of amino acids. And also you can see the different bonds that are formed between the molecule and the amino acids. Next, we did what was known as the boiling egg analysis. Boiling egg analysis can give you computationally an idea of how this molecule will work inside the system. And in the boiling egg analysis, you can check on how many of these molecules from Moringa oleifera can actually bypass the blood-brain barrier. How many of these molecules are limited to the gastrointestinal regions, etc. So now moving on to the importance of bioinformatics. Bioinformatics is important because there are a lot of scientific problems which are like very complex, and there are not many analytical solutions available. So you can use bioinformatics to address such complex problems. Nextly, you can also use bioinformatics for such experiments which are difficult to perform, which are dangerous, which are possibly expensive, or which are generally blind. So instead of running an analysis in the lab, you can possibly check for the activity computationally, and then based on the observations made computationally, you can run it in the lab. So that does not really make it blind after you have done a bioinformatics analysis. Bioinformatics is a growing area, and there have been a lot of debate about how we can use bioinformatics to replace a lot of experiments, most importantly, the animal studies. This cutting edge value of bioinformatics is now essentially being utilized as an alternative to animal studies. Then also bioinformatics can help you get a new dimension to the current experiments that you're being carrying out, and also this adds value to the research that you are already doing, and thereby it will be supporting the practical aspects of the research using a theoretical basis. And talking fast forward, bioinformatics is now being the major bridge or the hinge between biology and artificial intelligence and machine learning. But what are the limitations of bioinformatics? Since bioinformatics is a growing area, there is a debate over the accuracy of this particular technique or technology. So it is an ever-evolving field, and there is more advances that are needed to ascertain the findings that we are making using bioinformatics. So when this accuracy is debated, there is an obvious need for in vitro and in vivo validation of your in silico findings. And for you to do any of this, you need adequate expertise, and you definitely need a high performance computer. And more importantly, bioinformatics is developed based on theoretical concepts and algorithms. So it might not really be the similar result when you go into a lab-like condition or a physiological condition in vivo, because these theoretical concepts and algorithms might not necessarily be the same in vitro or in vivo. So with this, I would like to conclude this particular topic. Thank you all. And if you have any clarifications, or if you want to carry out any further discussion, you can always write to me, or you can contact me on WhatsApp. I'm open for discussions right now.
Hello, sir. Am I audible?
Yeah. Yes.
Okay. Thank you for the talk, sir. I am from a physics and computer science background. So I would like to
Okay.
know which languages are preferred for bioinformatics. Like, is Python more preferred or Fortran more preferred?
See, generally, the kind of languages that we work on in bioinformatics are Python, the R package, and the Perl. Okay, these are the
Okay.
common programs that we, me as a bioinformatics teacher as well. Uh, we constantly teach the students with these three programming languages. And when it comes to application of these languages, R programming is extensively used for analytical and statistical purposes, whereas Python is essential for development of these softwares or tools that you can use for bioinformatics analysis. Same with the Perl.
Okay. And for for Astro purposes, mostly Linux is preferred. So is the same case here as well in bioinformatics?
Linux is essential. Linux is OS. So Linux is essential to run the programming languages like R. But like if you have a Mac, I think Linux is by default there, and you can run your R programming there as well.
Okay. Thank you, sir.
Yeah.
Sir, I just have a clarification. So
Yeah.
let's say now that we are into computational biology, so
Mm.
is it possible to create new products out of the coding sequences of biomolecules? Is it possible?
Yes. Good that you asked me this question. I told you, I extensively work on Schrodinger. Okay, and
Yes.
in like I can give you an example of Schrodinger where you can use that particular suite. It's like it's a suite. It's not a software. It's a suite or a suite, whatever you call it. It's a package, and in Schrodinger, you can actually use it to develop formulations for cosmetics. You can these formulations, you can check for them. You can check how, like let's say you are developing a product for hair, like shampoo or hair serum or something. You can check on how exactly this product reacts with the hair molecules in the hair. You can also check on how this product or this formulation can interact with the packaging materials that you use. What is the shelf life? How exactly you need to store it, and everything. So there is scope. Like, I mean, if that is the kind of product you're asking me, biomolecular products, yes. But if you are talking about the computational products, definitely you can use bioinformatics with the help of programming to develop the computational products as well.
So in elaborating with the same, so just like whatever proteins that keratin proteins that you just said about, so
Hmm.
will it be applicable in the same in real time too?
Hypothetically, it is supposed to be applicable that way. In most of the cases, in my experience of drug discovery, we are working on a few molecules, and we have gone to the extent of testing them in animals, and we have confirmed their activity in silico. There is definitely the activity of this molecule in the animal as well. So in most of the cases, right, although it is not like 100% output, I could tell you 90% of the cases it works.
Okay.
Yeah.
Okay, sir. So I think that is all.
Yeah.
So well, thank you very much, sir. Thank you for it was really a very informative session today. And it was great to know and learn about the fantasies that computers could actually do to help biologists with whatever work you just said about. And pharmacogenomics itself is a broad area of study. And thank you for explaining about the drug discovery. And overall, I think this was a complete package for learning about all the libraries, databases, and the sites to explore the data and all of this. So it was really informative. Thank you very much, sir.
Yeah, you're welcome, Dhanashree, and thank you so much. Thanks for the Nakshatra team to organize this event, as I told you earlier. Thank you.